going to tell you a little bit about the work on microbiome in this field of sepsis. Some of that is from the lab, some is from others. And these are my disclosures, mainly grants that fund the kind of work that we do. And most of you have some understanding and some knowledge, or a lot of knowledge, about the microbiome, but it's basically the fact that we have recognized lately in a much broader dimension because of advances in technology that were inhabited by a large number of bacteria. Indeed, we have more bacteria cells than human cells. And if you consider the amount of genes that those bacteria contain, you end up with having many more functional genes from bacteria than functional genes from our own human DNA. So this tells us a little bit about the potential of what those microbes are, but basically exposes a fact that we knew for a long time, which is that microbes are not an isolation, but actually they are organized everywhere in complex communities. But so far, we have relied a lot on understanding that microbial world based on isolation methods that basically are tuned up to isolate one bacteria of interest. And quite commonly, this is biased towards bacteria that we think that have a pathogenic role, that we've known that have some pathogenic role. The advances in the last 20 years is that now we have culture-independent methods that rely on identification of bacterial products such as DNA, RNA, or proteins, and that allows to characterize the microbes in a much broader and comprehensive view of the microbial world. So if you think about this, we have moved away from just focusing on pathogens and how the host react to these pathogens to understand lung disease, to now start understanding that there is a complex set of microbes that affects the immunological tone, affects host susceptibility. And this is quite a bit of a focus of studies on areas where there's a lot of microbes, such as in the upper airways or cavity and in the GI tract. And there's a lot of investigations showing how these microbes, especially the microbes in the gut, are contributing to set the immune tone, the systemic immune tone, and through that systemic migratory of inflammatory markers can affect lung disease in what's called the gut-lung axis. In the last 15 years, we have also started recognizing, and that's what my lab focuses mostly, that microbes get into the lower airways, and actually they exert tremendous effect on the immune tone in the setting of health and the setting of disease. So that's kind of a general introduction of what the microbiome is, but why should we care about this? What does it bring in the field of sepsis, which is what I was asked to discuss? And as you know, there a lot of the patients that we admit with sepsis, they're actually culture negative. And this is just a representation of the percentage of culture positivity among people that have different type of sepsis. But what's relevant is that when you look at the outcome of those patients that have culture positivity versus those with culture negativity, they're very similar. They both have similar kind of clinical outcomes. So the presence of a culture positivity is nice because it provides you some guidance about some part of what's going on, but it doesn't provide a lot of guidance when the results are not there. And I'm going to tell you also that there's much more than what you just find in culture. So this is a recent paper from the group in San Francisco where they have profiled the blood for looking for microbial DNA in blood among patients that have different type of sepsis, especially substratified for those that have culture positivity, culture negativity. And what they find is a lot of the patients, even those that have culture negativity, they see bacterial DNA circulating in blood. But let me give you another example, also from the same group in San Francisco. And we're going to have one of the speakers closing the session with So maybe there might be some overlap, hopefully not too much. If you look at the lower airways of patients with lower respiratory tract infections, and you look at the results on culture and the results on these culture-independent methods, and here there are two culture-independent methods, one that rely on DNA, which is metagenome, the other one that relies on detecting RNA, which is metatranscriptome. You can see that there are patients that you can detect the bacteria, the same bacteria in both methods, as displayed by red. But there's this dark red group that it can only be detected by culture independent and not by standard culture methods. So it's not that one will replace the other, because there are also bacteria that it might be easier to detect in culture than culture independence, as displayed here. But it's just to point out the fact that there's much more in terms of if you just think about the pathogens or other potential pathogens, there's much more in those samples. But I'm going to take it one notch even further. It's not just pathogens. So why do I say that? So I'm going to use an example of a set of data that we generated. So we've been studying patients with viral sepsis, patients with SARS-CoV-2. These were the sickest of the patients that we were admitted at NYU and Bellevue. And when you look at the lower airways of these patients, and you look at the genomic material coming from microbes, including viruses, fungi, and bacteria, you see a complex network, a complex set of microbes, which are displayed here in what's called a co-occurrence network. So the proximity and the relationship between these microbes are displayed by this. And the color that you see in each of these symbols, which represent a unique bacteria, virus, or fungi, taxa, the color represents how likely or what's their association with poor outcome, in this case, mortality. So if you see them red, it means that these are positively associated with mortality. So you see here in the center, SARS-CoV-2. It's not surprised that SARS-CoV-2 is red, because that means that the more SARS-CoV-2 this patient had in their lower airways, the higher the risk for mortality. That's no surprise. However, what you also see is that there are many others, including bacteria and fungi, that are also red, that coexist in those lower airway samples of these patients that also are associated with poor outcome. So let me explain this a little bit further. So I told you, viral load in the lower airways, the higher is associated with mortality that we show in this paper. If you now measure not viral load, but bacterial load, you see that patients that had higher bacterial burden in the lower airways. And if you look at what specific bacteria is present there, you see a lot of these bubbles that are colored these are statistically significant bacteria that were associated with worse outcome. Okay, and you can look at the names, and I'm not pretending that you're going to be able to read all of this, but the initial impression was we wanted to look for, the usual suspects, the usual respiratory pathogens that we thought that maybe these patients were getting more susceptible to get a secondary infection with another respiratory pathogen. Turns out that if you look at the names, you're not going to see many of the classical pathogens. And this big bubble that you see there is actually a Mycoplasma salivarium, which is an oral commensal that was highly abundant in the lower airways of these patients. Which poses the question, what is the role of these other microbes? Are they contributing to the process? Now, this data is not on isolation. If I can show you a similar set, this is from the group of Netherlands, where they also look at the lower airway microbiome and the fungi in the lower airways of patients who are critically ill due to SARS-CoV-2. They show that both the bacterial burden and the fungal burden was associated with worse outcome. So we can start thinking that these other microbes, beyond the pathogen, beyond what we think is, the bad guy, there's other sort of microbes that may contribute to our ability to predict or to estimate poor outcome. So you can think about developing these kind of microbial signatures that predict with certain accuracy worse outcome. The good thing is that this can be broadened up, and you can also include host signatures that might also have different prediction power for poor outcome. And you could combine the two to try to develop even better biomarkers that will predict outcome. So if I go back to that paper from Langelier, I show you that they have microbial markers with metagenome metatranscriptome, but they also have hosts. They have host transcriptome. And they can show that different transcripts, different genes, upregulation of different genes was associated with the presence of infection. So they did just what I said. You can combine microbial signatures that have high prediction power with host signatures that have high predictive power. And by combining the two, you can develop a model with certain accuracy, in this case, for the diagnosis of lower respiratory tract infection. But in a follow-up paper, they did the same thing with the systemic samples on sepsis. So if you look at the microbial genetic molecular markers from blood plus the host transcriptomics, you can develop this combination of the two to have a pretty good predictive model, in this case for the diagnosis of viral infection or viral sepsis or sepsis. And when you look at what are the signatures in this case for viral infections, they see the usual suspects in the transcriptomic data, a lot of interferon activation that are associated with the diagnosis. In many ways, this is great, but is it going to be clinically usable to have a diagnostic? Do you need a diagnostic? I think it started to provide insights into what else is going on with this kind of unbiased approach. The other aspect that I've been dancing back and forth in this talk is that you can also try to use this as a predictor, a predictor of clinical outcome. In this case, going back to the viral sepsis cohort of SARS-CoV-2, we combine microbial signatures with the metatranscriptome, host signatures with the host transcriptome, all from the lower airways. And the goal here is to detect the purple dots, the patients that died. And you can see that the combination of the two provides good accuracy at identifying those and a good negative predictive value. You don't have any purple dots here. This needs to be validated. It's not that I'm trying to tell you that this is close to be a clinically usable biomarker, but you can start teasing out signals, because if you then look at the host transcriptome, you can start looking at what is that is going on in these patients that have poor outcome. They all seem very equally sick. However, we cannot predict what the outcome is, what's going on in this. And you start seeing pathways that are regulated differentially as compared to those that survive. And a lot falls into pathways that we already have some idea that this might be important, such as interferon gammas and mechanism to respond to RNA viruses. So this is a lot of associative data. There's some experimental data that I didn't go into it. There's not a whole lot of experimental data in the setting of sepsis. It's very difficult to do it in human, and the mouse work, the preclinical work, are usually relying on CLP models. But the general concept that we and others are trying to dissect is that this microbiome exists in us, exists in the lower airways, exists in the GI tract. It affects our immune system, and it's been affected tremendously by external factors. This is a way to regulate immune function, and abnormalities or disruptive microbiome can affect the immune system, making you more susceptible to a pathogen or affecting how the pathogen ultimately causes injury. And the other complex feature that to dissect in human studies is the fact that these patients obviously are exposed to a lot of antibiotics, which eventually disrupt the microbiome. So I think the idea is that we need to try to start dissecting this complex vicious circle that septic patients fall into, to try to identify what are the potential microbial interventions, what are the effects that current therapies have on the microbiomes, and can we promote a healthier microbiome that will either make you less susceptible to pathogens or cause or affect how the host is responding to them. So I'm going to just end up here, so we try to catch up a little bit on time, but I hope I convince you. One is that lungs are not sterile, and even when we look in blood, there's bacterial DNA. It doesn't mean that these are live bacteria, but there is bacterial translocation and bacterial signals present in there that might be important to understand host immune responses in the and either diagnosis or prognosis in the setting of sepsis. I think that uncovering the functional aspects of these microbes are going to be key to understand inflammatory damage and ultimately pathogen susceptibility and propagation of disease and organ damage. I just want to finish by acknowledging the many people working in the lab and the funding agencies that support this work. I'll be happy to take some questions later when you guys decide. Thank you very much.